Okay, we start. You need to be a bit more responsive today because I need to cover three things. It's just a quick summary and then the mistakes that you actually need to know. You need the MCQ marks. You really need MCQ marks. That's all I can say. So hopefully this one will clarify some stuff. Okay, the molecular genetics, right? Just again in a nutshell, so some of you will still be confused. Molecular genetics, right? We want to talk about two very important things, and the ultimate aim of talking about this is in a cell is to make the protein. In a cell, we are very concerned with making the protein, and making a protein actually has two steps. So the step number one is the transcription. Right? Step number one is the transcription. Step number two is the translation. Transcription is essentially within the nucleus, whereby when you have two DNA strands like this, we only use one. And this one that we are using for transcription, we call this the DNA template. After you transcribe what is on the DNA, you will get a new sequence. After you transcribe, you will get a new sequence. Okay, this one. Transcribe. All you need to know that during this stage here, right, when we are talking about it, this one is actually this stage here is actually number one. This one is transcription. After you transcribe the data, actually on the basis of the DNA template, you will get a new what do you call that? Hold on, let me change the color so you can see. Okay. So you will get to see a new color. Okay. Like this one basically is a new this is called the messenger RNA, right? So this new strand of the messenger RNA, I'm just going to change the color for those who need so that it's not confusing for you. It looks like a green color like that. Take note that this one is an RNA, and so the thymine in the DNA is replaced by the uracil. Okay, T replaced by the U. Okay, so this messenger RNA don't stay inside. You need to actually read it and read the language and translate it. Okay, so it has to come out. It has to come out of the nucleus. When it exits the nucleus, as you all know that this is actually the nuclear pore. Everybody knows that this is the nuclear pore, right? So after we are done with this, right, where does the mRNA actually goes? The mRNA will go and look for a very very important structure inside the cell, which we all know, and this structure is, it will look for the what? It will look for this thing. This is called the ribosome, right? And then it will just move the ribosome, and your mRNA will come in between the ribosome here. That's all you need to know, okay? So it will come to the ribosome. Ribosome are the cell structure that actually make the protein, correct or not? Okay, so when it comes here, then we need to start another process already, right? We need to start number two. Number two is translation. Okay, just now was number one, transcription. Remember, I also mentioned to you that every three of this, for example, there is a U, and G. This is the mRNA, okay? Every three of this every 3 bases on mRNA, you just need to say that every 3 bases on mRNA code for what? Code for an amino acid. A lot of you like to jump the gun. Straight away you will say it makes a protein. No, the protein not so easily made. It's huge. You make it from the most basic unit first. Make the amino acid first. So every 3 of them will code for 1 amino acid. Anyhow say so let's say this amino acid here, I have okay, I'm gonna change color because I think you cannot see it. Hold on the orange color. Okay, so this one, let's say this one is a P, all right, maybe one here, okay, then maybe here is another one, maybe here another maybe Z, and then it goes on, right or not? And you will realize that after you come here, you form a lot of amino acid, right? They will all join up. And this very long chain of the amino acids, right, it will make up your polypeptide. Ring a bell already, right? Then finally, when it has a 3D configuration or shape, it will then, okay, better right here, 3D configuration. Okay, this one I don't want okay, then you will get a protein. For example, you will get this, right? Okay, then you get a protein. Understand or not? It's a very simplistic view, but this is really all you actually need to know for your what do you call that, your molecular genetics. You just need to know the 2 words, you need to know the process. So essentially, let me summarize again what they are actually is. Transcription is basically one DNA template, guys. Okay, it will actually transcribe and give you mRNA. Translation is mRNA go to the ribosome, then every 3 base code for 1 amino acid, then form polypeptide, then it will form finally our protein. I know you studied, everyone, but the extent and the depth of how you actually study is also very important. We can't always give sweeping statements. So when you study this particular topic, it's okay to be general, because this part here is too difficult. All you need to know is in this slide. You either refer to the notes I given to you, or you can look at this one, you withdraw one set of your own and you add in a little bit from the other side. Then that will be all you actually need to know. Under molecular genetics, the part about the genetic engineering, you're very, very good with it. So I'm not going to go through that part. I'm only going through this one only. Okay? So this is really the super summary of it. This is all you need to know. Okay, you have to ask questions. If you have anything, don't ask me after that 2 hours later, you probably forget what I just said. Okay, can you raise queries now? I'm down in the map already. This is all for the confusing part. Anybody?
Mr. Tan, can you explain the structure of DNA?
DNA, can, wait, I add in one more word first. Amino acid. Okay, so whatever it takes, only those things I said here are important for this part. Structure of DNA, when somebody asked me, you did wrongly in the essay. I haven't reached the essay part yet. A lot of you didn't do the essay part for DNA structure. Why? Anybody want to tell me why? You just asked me the day before. You never do the essay. Oh, I know already. You all do the all question. You say everybody go and do the genetic engineering. Yeah. Okay, structure of DNA, right? Oh, I prefer to do it. I prefer to craft an answer from micro, small, then I will go to macro. I find it easier to write. And I suggest you do it this way. If you start writing the answer from double helix, after that you find it's very difficult to go all the way down. So a few points that we want to take note. Number one, basic unit of the DNA is always a nucleotide. Am I right? So you don't have to draw. You just need to list the nucleotide. Will have a. You must state clearly that it's a deoxyribose sugar. So if you write that it's a sugar, then it's not good enough. It must be deoxy because this is DNA, deoxyribonucleic acid plus a phosphate group. And we also have a nitrogenous base. Do not just stop at the base. This is a nitrogenous base. So the important sharpen, the precision is here. Okay, sorry. Nitrogenous base. So the precision involved here is really must be very precise one. The deoxyribose sugar, the nitrogenous base. Don't have to talk about the bases first. You wait. This is the smallest unit, right? Isn't this the smallest unit? So you remember now after that, I have the smallest unit already. I need to join it up. So this is the joining. Okay, so how? Many nucleotides join to give a very important word, polynucleotide. So it simply means if one of them is like that, and then you got the P here, right? And then you got this other base here, then it will join, give you a very long chain. Correct or not? This will give you one polynucleotide. So far okay? You need to get the meaning of it. Instead of trying to memorize word for word, you have to understand the meaning, how I craft the answer. Then the words will come into place. Okay, ready? There's too much to remember. Okay, then one, then you know a DNA actually it's not made up of one only, it's made up of two. Okay, then you need to get on number 3. Alright, one DNA molecule is made up of what? Made up of what? 2 polynucleotide chains. Am I right? Here I can insert one more word, which is the direction of the chain, which are, see I add in here, anti-parallel to each other. Then I was for the third one already, right now. So again, important stuff here, anti-parallel, right? And we also have the 2 polynucleotide chains. Okay, so done with this one already. Then you need to ask yourself how they join. You didn't say so must say they join by what? The rule of complementary what? Base pairing. Right now. Whereby, then you must state the rule, don't stop there. Whereby A binds to T, C binds to G. Then complete already. So the important thing to remember here is the rule of complementary base pairing. Okay, are we done? Not exactly, not really. Are we really done? Only like that, right? Correct or not? Then how? What is actually the missing one? Nobody know. Hello? Class. Hey, what's the last one? Class.
Double helix.
About the double helix structure.
Yeah, so still dare to say some of you stop here then never say, very weird after that here, then after that you will say that what the two strands will twist and turn to form a double helix. So important word is this one, right? Okay, right, about 5 already. 5 marks. If some of you, if you like, you can add in one more bit of details here. Also, you can say that between the bases, they are bonded. They are binded by what? Hydrogen bonds. If you don't find enough points to write lah. Cannot. Actually, like that will give you a pretty good answer. It's a very easy answer to remember. So write from small and then to big. Something that we always advocate, easier to write. Questions, anyone? This one?
Mr. Tan.
Uh-huh.
Do we need to write about the sugar phosphate backbone?
Can also, if you want, if you find maybe not sufficient, but where do you want to insert sugar phosphate? This is how you look at your answer. Where you want to write sugar phosphate? 1, 2, cannot be 1, 2, 3, right? I think.
Between 4 and 5.
Yeah, maybe 4, 5. So let's try put in the sugar phosphate backbone. Wait, the two strands will twist and you're right here, with constant means doesn't change, sugar phosphate backbone. So the purple color are my additions after I craft the first answer. When you write the first answer, right, you leave a bit of space at the bottom if it's an essay usually. See how much things you want to add in or not. Do not finish the whole question, and after that then suddenly you go and tell me that okay, the whole answer you don't want, right, and you start cancelling. Our class don't have the issue so it's not so bad. So plan the answer, leave a bit of space after that, then you can actually insert and see how. Anything else you want to add? This one will score full marks for 5 or 6. The 6 mark I didn't break it up, but if I want to break it into 6, it's possible.
Mr. Tan, instead of saying with a constant, can we say supported by a constant sugar phosphate backbone?
Can, supporter is the same. That means it's always in the center, so it doesn't change lah. The only thing that changes is the bases. Also can.
Okay, thank you.
Any more? So genetic engineering you want go through all the one is like very good already. So not so much I actually need to run through it, that one, Can, no sound means okay, we go next one. Okay, I stop this one first, alright?